Okay, we're doing Chapter 10, Molecular Biology of the Gene, which means we're going to be looking at DNA and RNA at their structure and at the proteins that help them out. DNA, pretty cool. It's a very simple molecule, basically repeating patterns. But because you can have these repeating patterns, they were actually able to create a computer using DNA, which I think is pretty awesome. Instead of the typical yes, no computer, they were able to use the repeating base pairs of DNA. So because DNA is so simple, there were people who thought that DNA could not possibly be the molecule that was the genetic factor in life. Life was far too complex. It had to be a more complex molecule, and there were a good number of people who thought that that should be a protein because protein molecules can be very complex. And Hershey and Chase designed an experiment to figure that question out. Is it the DNA or is it the protein? And they chose to work on something called a phage. A phage is just a virus that's going to infect a bacteria, and it's a really good choice because a virus is only a protein coat, right? The protein coat and the genetic material, the DNA on the inside. So it's pretty simple to work with, and then they could figure out, was it the protein coat or was it the DNA? Okay, so the Hershey-Chase experiment, what they do is they take the phage, the virus, and in one trial they make the protein coat here radioactive. And they follow that and they see if the radioactive material goes into the bacteria. So the way they do that is they throw it into a blender right here. You'd be surprised how much biology is done by throwing something into a blender. And when they take that material and they centrifuge it, what happens in the centrifuge is the lighter phages, the viruses, end up in the liquid up here, and the bacteria are heavier and they go to the bottom and it becomes a pellet, all right, a nice dense pellet, and they see where the radioactivity is. And in this case, the radioactivity, the radioactive protein, was still with the virus, even though the bacteria had been infected. So they knew that that was not the genetic material. In the second trial, they take the DNA here and they make the DNA radioactive. That gets injected into the bacteria. They throw it in the same old blender, blend it up, and they look in their centrifuge. They centrifuge it. And this time, the bacteria here become radioactive. That genetic material that infects the bacteria has gone into them. The phages are no longer radioactive. The protein coat stays up here where the phages are. And so they figured out that DNA is in fact that inheritable factor, that what passes your genes on or what can infect things is in fact DNA. Alright, so what is a phage reproductive cycle? It's pretty simple. You have your little boy, they look like alien invaders here, right? Lands on the poor bacteria. It's going to come over here. It's going to inject the DNA. Now this bacteria stops doing whatever it was doing, whatever it was making for itself, and it starts making viruses. And when that is complete, it explodes and releases these viruses to go infect new bacteria. Alright, DNA and RNA, here's the structure of them. They're made of long chains of nucleotide monomers, right? And we remember that. Here's your backbone. It's a phosphate to a sugar, to a phosphate, to a sugar, to a phosphate, to a sugar. And you have your nitrogenous base pairs going down the middle of it. Phosphate groups are very common. Here's a picture of a phosphate group. You use it in like ATP, those kind of things. So these are common in biology. And so are your sugars. This is the deoxyribose. This one's going to be DNA. And then here's your nitrogenous base. Okay, DNA has 4 different nitrogenous bases. It has thymine and cytosine, which are pyrimidines. Those are single rings. See, it's only one ring here and one ring here. All right, then you have the purines, which are adenine and guanine, and those are double rings. So they're going to be twice as big. And so what you always have in DNA is a purine always binds to pyrimidine. And so you always have 3 rings going across. You'll have adenine here binding to thymine, remember A to T, and you'll have cytosine bonding to guanine, G to C, if you remember, and that will always give you 3 rings across your DNA, so it never bulges or gets skinny. It's always 3 rings across. All right, RNA, very similar, except that now instead of deoxyribose, we have just plain old ribose, so the sugar's slightly different. We have our same old phosphate group, and we have uracil instead of thymine on RNA. Okay, so the structure of DNA, it was basically a race. People knew it was DNA after Hershey-Chase, and people wanted to figure out the structure of it. There was a woman called Rosalind Franklin who was doing X-ray crystallography on DNA, and Watson and Crick were able to see her imaging, and from that they were able to figure out the 3-dimensional structure of DNA, and obviously they won the race. Everyone remembers Watson and Crick. Alright, so the basic structure of DNA, kind of like a ladder, you have, this would be where your nitrogenous bases are, and you would have your phosphate sugar backbone going down the sides, and then it's twisted. It's a double helix. Alright, so what holds this together is hydrogen bonding, right, between A and T. You can see here, you got this holding it together are your hydrogen bonds between A and T, and between C and G you get 3 hydrogen bonds, holds the ladder together basically. Alright, so how does DNA replicate? Well, the beauty of DNA is A and T always bind together, and C and G always bind together. So when you unzip your DNA here, right, you always know what pair to stick in the middle, because you can always only have a T going to the A, and on the other side of that unzipped ladder, you will have an A bonding to a T. You will always have the G to the C, and on the other side of your unzipped ladder, you're going to have a C to a G. It is absolutely a beautiful system. And so then you have 2 daughter pairs that are identical to the original DNA. Okay, so DNA replication, the process becomes complex because it needs proteins to unzip it. The theory is simple, the A to T, the G to C. But before you can replicate it, you have to untwist it and unwind it and unzip it to open up those base pairs to have access to them. So we're going to use a couple enzymes to do that. Okay, so DNA, as it replicates, it is going to bubble or open up at a site of replication, and the daughter strand is going to start to replicate. So here you go, you're getting a daughter strand. Here's a bubble. Here's another bubble. These expand out, and then you have an enzyme called DNA ligase that's going to zip these strands together. So what you have at the end is you have one parent strand here, the original DNA, and you have one daughter strand attached to that. They call this semi-conservative. What they mean is that with the new strand that you have, one was an original parent and one's the daughter strand. And here you have the same thing. You have one original strand with you, and you have one new strand. Now, each strand is always oriented opposite. So you always have one going 5' to 3', and you have one, so here's your 5' to your 3', right? And you always have the one it's attached to is 5' to 3' the other direction. And that matters with replication because DNA always replicates in a 5' to 3' fashion. It always goes one direction. So here you are, and you've unzipped your DNA, and you're starting to replicate it. And when you're over here, you can see you can make a nice 5' to 3' new daughter strand. But on this side, as it's going from 5' to 3', you end up with these little short strands, all right? And what will happen is the ligase, again, will come zip this together. So on one side, you get one easy strand. On the other side, you're going to get these little short pieces. Okay, so DNA, what it does is it codes for proteins. So remember we talked about proteins being the more complex molecule. And DNA codes for the sequence of polypeptides. It's going to give it the primary structure to the DNA. And that is why you can have such a simple molecule take control of such a complex thing such as life. By changing the sequence of base pairs, we change the amino acid sequence, which changes the protein, and all these different proteins go out and do all these different functions. But it's the DNA that ultimately codes for those proteins. All right, so the way that works is we're going to talk about transcription, which is where we're going to take DNA and make it into RNA. And then we have translation, which takes RNA and makes it into an actual protein. And I think of transcription, you can use whatever trick you like, but I think of like transcribing from one language to another. You're going to take DNA and you're going to do transcription into RNA. Translation makes it into a whole different thing. It makes it into a protein. All right, so they didn't know like how long of a piece of DNA you could use, or you needed to have to make an enzyme or a protein. Was it 1-to-1? Did you have 5 genes make a protein? They didn't know. And they actually used bread mold and figured this out. There was a bread mold that couldn't grow on a specific substance. It was missing an enzyme. And they were able to show that it was missing one gene. And so that really kind of proved to them that one gene controlled one enzyme. It's that one gene to one protein concept came from a bread mold. All right. So DNA is the language of our genetic information, and the way it's written out are in these triplets of codons. So we're going to take DNA, the A to T or the G to C, and when we have 3 of them, a triplet, we have the coding for an amino acid. All right, so here's a quick summary of transcription translation. Here you have a DNA molecule over here. And usually you do one gene at a time, so it's gene 3's turn to be to have transcription and then translation to be made into a protein. You unzip the DNA strand. This is basic transcription. You make your RNA sequence off of the DNA strand. Every 3 RNA nitrogenous base pairs codes for 1 amino acid. So you link your amino acids together. That's the primary structure of a protein. That's your polypeptide chain, and that sums it up. Don't write this down. This is in your book, page 192. So don't write this down. This is just the genetic code for your amino acids, and the way you read this is you start with your first base on this side. So if you have a U as your first base, and a U as your second base, and a U as your third base, you are coding for phenylalanine. If you are an A as your first base, maybe a C as your second base, and a C, you're over here for this amino acid. And this is in the book, so don't write this down. You can just look this up. All right, so let's look at translating the genetic code. This is similar to that first slide, which I said summed it up. You take your DNA, that's going to be transcribed right here. You unzip it, okay? And so T binds to A, right? And then A binds to T for DNA. Well, when we go to RNA, remember we substitute in a uracil because there is no T in RNA. And once you have 3, an AUG is always your start codon. You might as well just memorize that one. It's always going to be your start codon. No protein's going to start without AUG. And then you have your next triplet, You go through translation, and you make your polypeptide. All right, transcription is what's going to produce the genetic material in the form of RNA. So what's transcription? We have our RNA polymerase. That's our enzyme that's going to go through, and that's one of our protein helpers in nucleic acids. You have your RNA. So this whole thing represents RNA polymerase. We have the DNA strand going through it. We have the direction of transcription. Remember, it's always going to be 5' to 3'. And we're going to add in our base pairs here. All right, so in the nucleus, the DNA helix unzips. Your RNA nucleotides line up along your DNA, going with the base pair rules, right? And you get a single-stranded messenger RNA that peels away from the gene. The DNA strand then rejoins, and it stays in the nucleus. The cytoplasm of the cell is a big, scary place where RNA and DNA can get degraded at any minute. We do not put our genetic code, our DNA, out into the cytoplasm. We send the messenger RNA. Who cares if it gets degraded? You can kill the messenger. It's not a big deal. The original DNA will stay safe. So the messenger RNA is going to go out into the cytoplasm and get translated into a protein, but the DNA stays in the nucleus. So here's your transcription. First you have initiation, and that means that the RNA polymerase binds on to the strand, the DNA strand. You have elongation, and that's the process of doing this base pairing, right? We're just making a long RNA strand now. And finally, it hits a termination sequence. The RNA polymerase falls off the DNA strand, and you have a completed RNA strand. This is all still going on in the nucleus. We haven't sent the messenger RNA out into the big scary cytoplasm. The last part of it is your introns and exons. The introns get removed, and the way I remember that introns get removed is I think of them as interruptons. It interrupts the sequence. So we're going to splice them out. The exons are actually the ones we keep. The exons get spliced together so that you have one final code, and then that gets sent off, and actually you put a tail and a cap on it before you do this, and then that gets sent out into the cytoplasm. One thing the tail and cap do is it protects it before, as it gets sent out into the cytoplasm.